Hello everyone, welcome to today's video on glycolysis. The whole purpose of having food is to gain energy. But to gain energy from the carbohydrate, it has to go through the process called glycolysis. Glycolysis is a 10-step process and it is mainly divided into two phases, that is energy requiring phase and another is energy releasing phase. It is just like an investment. To gain more, we have to invest at least something. So here also we invested a little to gain more at the end. Glycolysis looks difficult to understand at first, but trust me, if you understand each step, why it is happening and what are the reasons for it, then it will be very easy to understand and then you don't have to mug up all those chemical names. It will be very easy to memorize. So let's start with the first step. When glucose molecules enter into the cell to generate energy, it enters into the cell through the GLUT protein. Well, there are various types of GLUT protein and we already saw them in detail in the previous video. And if in case you missed that video, I will provide a link in the description box so that you can see that. So this is a GLUT protein which provides a facilitated diffusion. And you know that in facilitated diffusion, the transportation takes place from the higher concentration to the lower concentration. Because of this, there is a chance that glucose can escape back into the blood circulation, which we don't want. We want that glucose molecule to stay in the cell to generate energy. That's why glucose molecule changes its structure by adding a phosphate molecule to it. And it happened by the process called phosphorylation. And it is carried out by the enzyme glucokinase or hexokinase. So what are these two enzymes? Well, the glucokinase is the enzyme found in the liver, while hexokinase is found in all the cells of the body including liver. Glucokinase have low affinity towards the glucose. It means glucokinase comes into action when there is a high glucose concentration, while hexokinase works in lower concentration as well. And this is very helpful because glucokinase is found in the liver and store glucose in the form of glycogen, which leads to the fatty liver in the long period of time. On the other hand, hexokinase helps to utilize glucose for energy by the muscles and various organs and all the different chemical processes that's continuously going on in the body. That's why your dietitian also recommends to have low glycemic food, which will not spike up your blood sugar level. So hexokinase will work more and eventually it can prevent the fatty liver. Okay, coming back to the glycolysis. So we were discussing about the phosphorylation. So what is phosphorylation? Phosphorylation means adding a phosphate group. But from where the glucose will get the phosphate molecule? It will get the phosphate molecule from the ATP. ATP is stand for the adenosine triphosphate. Tri means 3. So these enzymes help in giving this one phosphate molecule out of this 3 phosphate from the ATP to glucose. So by giving one molecule to glucose, it converts itself into ADP, that is adenosine diphosphate. Di means 2. And as this phosphate molecule attached to the 6 carbon atom of the glucose, the name of the product is glucose 6-phosphate. And as we are adding a phosphate molecule to it, this process is irreversible. Now coming to the second step. In the second step, this glucose 6-phosphate molecule changes its structure into fructose 6-phosphate molecule. And this process is called isomerization. As here the formula will be the same, that is C6, H12 and O6, and only the structure is changing. The name of the process is isomerization. In the isomerization process, only the structure will change but the formula will be the same. And the enzyme used here is phosphoglucoisomerase or phosphohexoisomerase. As the name suggests, as there is a one phosphate molecule, one glucose molecule, and isomerization is taking place. So the name of the enzyme is phosphoglucoisomerase. But what is the need for this isomerization? Can't we take this glucose as it is in the next step? The answer is no, we can't. Because in the third step, we are going to add one more phosphate molecule at the first carbon atom. But this is not possible with the glucose molecule, as there is an aldehyde group on the first carbon atom. That's why we are changing its structure into fructose 6-phosphate molecule. Because at the end of the glycolysis, what we want? We want to break this molecule, big 6 carbon molecule, into 2 small molecule of 3 carbon in each. But this is possible by adding one more phosphate molecule to make it unstable. That's why isomerization is a very important step. And as there is just isomerization is taking place, the second step is reversible. And now coming to the third step. In the third step, we are again going to add one more phosphate molecule at the first carbon atom and convert fructose 6-phosphate into fructose 1,6 bisphosphate molecule. The name 1,6 because the phosphate is attached to the first and the 6th carbon atom, and this process is catalyzed by the enzyme phosphofructokinase 1. Whenever the name comes kinase, it means either we are adding or taking out a phosphate molecule. And this time, from where we will get the phosphate? Again the same, that is from the ATP. We are taking one phosphate from the ATP and converting it into ADP. So up till now, we invested 2 ATP. This is again an irreversible process as we are donating phosphate molecule. And this is very important step in the process of glycolysis because it is also a rate limiting step. It means it will decide how fast glycolysis takes place, how fast the energy will generate. As here the enzyme PFK1 used, and the action of the PFK1 is regulated by the enzyme PFK2 and the hormone insulin. So what is this PFK2? The PFK2 is stand for enzyme phosphofructokinase 2. As the name suggests, it adds a phosphate molecule to the second carbon atom instead of the first carbon atom. But by adding a phosphate group at the second carbon will not help in the process of glycolysis. But this high level of PFK2 will activate PFK1. In the simple words, when the body is well fed, like right after the meal, the blood glucose level goes up and the pancreas secrete insulin, which activate PFK2, which result into more formation of fructose 2,6 bisphosphate molecule. And this high production of fructose 2,6 bisphosphate stimulate the production of PFK1 enzyme. So more PFK1 means that the glycolysis speed up and more glucose it converted into energy. Now when the body is in fasting state, like few hours after a meal, blood glucose level goes down and the pancreas now secrete the glucagon instead of insulin. Glucagon inhibits or slows down the PFK2 and resulting into less fructose 2,6 bisphosphate molecule, which inhibit PFK1 production. That slows down the glycolysis process. So this reaction is considered the rate limiting step of the glycolysis. It means how fast PFK1 converts fructose 6-phosphate to fructose 1,6 bisphosphate determines the speed at which all glycolysis happen. I hope you understand all this. I tried to explain as simpler as I can. Okay, coming to the fourth step. As by adding one more phosphate, this 1,6 bisphosphate is now an unstable molecule. That's why it breaks down into 2 molecules in the presence of enzyme aldolase. And this gives out 2 molecules, that is 3 carbon in each. One is glyceraldehyde 3-phosphate and another is dihydroxyacetone phosphate. The glyceraldehyde 3-phosphate is directly used in the glycolytic pathway. On the other hand, dihydroxyacetone phosphate is not able to use in the glycolysis because it has an acetone group, and so it need to be converted it into glyceraldehyde 3-phosphate molecule so that it can be used in the glycolytic pathway. And this is carried out by the enzyme triose phosphate isomerase. Here again, look at the name of the enzyme. Triose means 3, phosphate means phosphate molecule, and isomerase means change the structure but not the formula. So ultimately by this process we will get 2 molecules of glyceraldehyde 3-phosphate. So till this day it is considered as the energy requiring phase as we use 2 ATP. And now we are entering into the energy releasing phase, because here we will generate energy. So let's see how it will happen. In the next step, we are adding one more phosphate molecule to glyceraldehyde 3-phosphate. But this time we are not taking phosphate from the ATP. So here there is no kinase enzyme, but we are taking phosphate molecule from the inorganic phosphate which is present in the cytoplasm. So we will get 1,3-bisphosphoglycerate molecule, as here the phosphate molecule is attached to the first and the third carbon atom. And as there is again 2 phosphate molecule, so the name is 1,3-bisphosphoglycerate molecule. And this all happened by the enzyme glyceraldehyde 3-phosphate dehydrogenase, which creates 2 molecules of 1,3-bisphosphoglycerate. But why the name of the enzyme is dehydrogenase? Because here the glyceraldehyde 3-phosphate is undergoing oxidation. And oxidation means either removing hydrogen atom or adding oxygen molecule. And here we are removing hydrogen atoms. So the name is dehydrogenase. But how does it going to happen? Let's have a look into it. Here NAD plus is converted into NADH. And this reaction is very important to know, because here NAD plus actually steals one hydrogen atom from the glucose molecule. And you know, a hydrogen atom has a central positive proton and one negative electron which is flying around the proton. In other words, the proton forms the nucleus and the electron orbits around it. So this makes it neutral, means no charge at all. But if NAD plus steals hydrogen atom from the glucose, it comes in neutral. So the positive charge of the NAD is as it is. But here we need NADH without any positive charge. But what we are getting? NADH plus. So we have to remove this plus sign. But how can it happen? Well, the NAD plus actually steals 2 hydrogen atoms, but one complete, and from another hydrogen atom it steals only negatively charged electron and leaving behind positively charged hydrogen proton. So this negatively charged electron and positive charged NAD being neutral, and we will get NADH along with the positive charged hydrogen as a byproduct. And so this process is called oxidation, and by this way we will get NADH. So finally what we got from the 2 molecules of the glyceraldehyde 3-phosphate? We got 2 molecules of 1,3-bisphosphoglycerate, and along with this 2 molecules of NADH and 2 hydrogen atoms. In the upcoming videos we will see the usage of the NADH and the hydrogen atom in the cellular respiration. But for the time being, just keep this in mind. Okay, so moving forward, we got 2 molecules of the 1,3-bisphosphoglycerate. But now we want to get rid of this phosphate molecule. Do you remember in the energy requiring phase from where we got the phosphate? Yes, right. We got phosphate by utilizing ATP. And this is the time we will generate ATP by giving one phosphate to the ADP and converting it into ATP. But as here are 2 molecules of the 1,3-bisphosphoglycerate, we will get 2 ATPs, and we will get our product, that is 3-phosphoglycerate, and this happened by the enzyme phosphoglycerate kinase. In the next step, this molecule is rearranged and the phosphate attached to the second carbon atom from the third carbon atom. So this called as a phosphoglycerate, as called as 2-phosphoglycerate, and this happened by the enzyme phosphoglycerate mutase. So the question comes to mind, why mutase? Why not isomerase? Well, the mutase is also type of isomerase only, but here we are moving the functional group. In other words, we can say an isomerase is a general term for an enzyme that changes the form of a substrate without changing its formula. And a mutase is an enzyme that moves a functional group, such as phosphate, to a new location. That's why Here the names of the enzyme is phosphoglycerate mutase and not phosphoglycerate isomerase. Okay, moving forward to the next step, that is the ninth step, is the conversion of the 2-phosphoglycerate to 2-phosphoenol pyruvate by the enzyme enolase. So in this we are adding one oxygen between carbon and phosphate molecule and creating a one double bond between the second and third carbon atom. Otherwise in the whole process there is only single bond. Only here at the first time double bond come into picture. Here you can see a new name, that is enol, in phosphoenol pyruvate. This enol is a combination of two words, that is alcohol and the ethane group. So combining the two names, it gives the name enol. And along with this phosphoenol pyruvate, we will also get H2O, that is a water molecule, as a byproduct. And this is a reversible dehydration reaction. And as there are 2 phosphoglycerate molecules, we will get 2 molecules of the phosphoenol pyruvate and 2 molecules of water. So we are now at the last step of glycolysis. And now we are going to convert this phosphoenol pyruvate into pyruvate molecules. How? Obviously by removing the phosphate molecule. And how to remove this phosphate? Again the same, by donating. And this time donating phosphate molecule to the ADP instead of glucose molecule. So by this way convert ADP into ATP. And now as there are 2 pyruvate molecules, so total 2 ATP generated. And ATP is nothing but the energy. So what we are getting at the end of the glycolysis process? We are getting 4 ATP, 2 NADH and 2 pyruvate molecule. But as we are investing 2 ATP in the energy requiring phase, so by deducting that, finally we will get 2 ATP instead of 4 ATP and 2 NADH and 2 pyruvate molecules in the energy generating phase. So this is all about glycolysis. Thanks for watching till the end.